Okay, so genes and mutations. DNA molecule can be enormous. The bacterium E. coli has just one DNA molecule, which is 4 million base pairs long. There is enough information to code for several thousand proteins. The total DNA of a human cell is estimated to be about 3 multiplied 10 to the power 9 base pairs long. However, it is thought that only 3% of this DNA actually codes for protein. The function of much of the remainder is uncertain. This is just for your information, since you are a student of biology. A part of a DNA molecule, the nucleotide sequences for just one polypeptide, is called a gene. And one DNA molecule contains many genes. A change in the nucleotide sequence of a gene which may then result in an altered polypeptide is called a mutation. Most genes have several different variants called alleles, which originally arose by the process of mutation. Okay, these are actually something to learn by heart. I will explain to you. First of all, I need to explain you. I explained you before. You will know that the monomer for DNA is nucleotide. A group of 3 nucleotides, triplet nucleotides, produces an amino acid. A group of nucleotides is called a gene, which produces a polypeptide. A group of genes is called a DNA. A group of DNA is called a chromatin. 2 chromatins are joined together by centromere to form a chromosome. Okay, so now we also have known that in case of proteins, the monomer of protein is amino acid. 2 or more amino acids joined together to form a polypeptide. 2 or more polypeptides joined together to form a protein. Okay, now let's see. Say for example, this group of triplet nucleotide, adenine, and adenine, produces serine. This is not the exact combination for serine. There is a DNA chart which I will show you, where you can see to know the exact combination for the specific amino acid. Okay, so say for example, for any cause, this adenine was replaced with let's say cytosine. So adenine, cytosine is not the combination for serine. Say for example, it would produce proline. So this change in one DNA, one nucleotide, which caused an altered amino acid to be produced, is called a mutation. Now, a protein is very specific. I told you that the specific combination of amino acid is the characteristic of a protein. So since instead of serine, proline was produced, the function of the protein will be changed. Okay, we have learned what is mutation. Now let's see what is the meaning of alleles. Say for example, let's say that the protein responsible for skin color, white, was serine. And that time the nucleotide sequence was adenine. But for some reason, mutation happened, and the combination of nucleotide changed to adenine and cytosine. So proline was produced. Now say that since proline was produced, the function of the protein changed, and that it caused the skin color to be black. So this is one gene, and only one nucleotide was changed. For only one nucleotide being changed, the function of the polypeptide changed, but both function are as to give our skin a color. But adenine produces serine, which produces, say for example, I told you, white skin, and adenine and cytosine produces proline, which was for black skin. So the genes that contains adenine, and the genes that contained the part of the gene that contain the nucleotide adenine, cytosine, these 2 genes, because none of the other nucleotides in the genes was changed, so these 2 genes are called alleles of each other, because they work in the same sector, but their output is different. One produces white skin, one produces black skin. So this is the meaning of alleles. Okay, so you guys may have one confusion. I said that 2 chromatins makes up a chromosome. What I meant to say was that in nuclear division you will see that the chromatin fiber condenses into chromatid, and 2 chromatids are joined by centromere to form a chromosome. This is the most specific definition.